Well, thank you for having me. My name is Russ Tobe. I run programs for individuals with autism spectrum disorders at the Nathan Kline Institute, which is a research center in Orangeburg, New York. And I'm going to be talking about pharmacotherapy and autism spectrum disorders, going through some updates, as well as talking about some new frontiers and some initiatives that are going on in medication development. Before I get started with that, I wanted to talk about my role a little bit more, and specifically pertaining to medication development. So when we develop medications in psychiatry and do clinical trials, that's an industry collaborative process. The federal government doesn't put much funding support into new medication development and therapeutics, and so we do that in partnership with industry. And so I'm an employee of the state of New York, but I do receive research funding from several pharmaceutical companies in that collaboration. And so those pharmaceutical companies are Axial, Janssen, and Roche Pharmaceuticals. And I will be talking about some of the initiatives that we have going, but I'll be identifying those associations with those companies as I'm talking. But I just want to make people aware of that as we're moving forward. And generally, what I'll be talking about, since it's new medication development and new frontiers, these are not approved therapeutics at this point. These are therapeutics under investigation. But first, we're going to talk about some of what we know about autism spectrum disorders with respect to the biology and symptoms that some people can experience. So both autism spectrum disorder and intellectual and other developmental disabilities all present in early childhood. And so they are all developmental conditions and generally have a negative trajectory in one of several potential domains, including social development, intellectual, or emotional development. What we know about, though we don't know all the specific genetics and factors that necessarily drive ASD and intellectual disabilities and other developmental disabilities, we do know that they're incredibly biologically driven. However, most cases are idiopathic. We can't necessarily identify a cause. But it's important to think about those biological factors for several reasons. One is that these are among the most heritable disorders that we have in medicine, period, not even psychiatry. So arguably more heritable than things like diabetes, hypertension. And so it's important to recognize that from an advocacy perspective. We have a tremendous amount of family stigma, and that stigma comes from dating back to the 1940s when Lee O'Connor was speculating on the idea of the emotional availability of the mother to the child as they're developing, and wondering if a less emotionally available mother may be a factor in development of developmental disabilities for individuals, specifically in autism spectrum disorders. He later, after data came in and additional information, pulled back from that position and overtly rejected it. But some of the damage had been done. There was another individual, Bruno Bettelheim, who really put forward this concept of the refrigerator mother, this sort of unavailable, emotionally unavailable mother, and that's the driver of autism spectrum disorders. And in fact, the intervention was then to separate the child from the family, which is, of course, the last thing we would think of doing in the modern era. But we still carry that concept of the refrigerator mother, the emotionally unavailable parent, as a driver of ASD, and it's just not the case. There's no data to support that at all. But that family and community stigma continues to some degree, and that can limit access to early intervention services, which are the one thing that we know really can help individuals who have developmental disabilities get more on a good trajectory and a forward-moving path. So that's just, of course, important to keep in mind that history. These are highly prevalent conditions. In any given classroom setting of approximately 30 or so children, one of those children, on average, will have an autism spectrum disorder or developmental disability. ASD is a little bit more prevalent than intellectual disability, but they're both quite common conditions. Contrary to popular belief and what most people understand in the lay community, these are actually, though they are co-occurring conditions with each other, really only a minority of individuals with intellectual disability have autism spectrum disorders and vice versa. And so the data are still in debate, but probably about less than a quarter of individuals with autism spectrum disorder have intellectual disability. And the reason that people often think that intellectual disability is highly co-occurring with autism spectrum disorders is based on old data. So if you actually look in community populations carefully, many of the individuals with ASD are really not identified. And so in older studies, clinic populations were investigated. And so, and children in clinic populations tend to have different types of symptom presentation and expression of their condition than children who are in community-based settings. And so, so that's important to also keep in mind that when we're thinking about therapeutic interventions, both psychotherapeutic, environmental, medication, biological intervention, we have to recognize that there's a real diversity with a number of different strengths within this population of individuals. And the other thing to keep in mind, too, is that both conditions have very high co-occurrence rates with other types of disorders. Some examples include mood disorders, tic disorders, seizure disorders, and other types of more severe persistent mental health disorders, such as psychotic illness. Those conditions are all relatively rare, but they're at much higher community prevalence rates within ASD relative to the sort of typically developing community. These are some of the symptoms that we tend to see for individuals with autism spectrum disorders. These are diverse symptoms, and anybody can have any sort of blending of these symptoms. So every individual with ASD is unique, but the symptoms cluster into two main domains, and these are what we consider the core domains of symptoms. And it's important to remember core because we don't have therapeutics that are developed to target core, at least on the medication side. So the first core bin are deficits in social communication and interaction. That can include how people relate, how people reciprocate in conversation, their back and forth capacity, and then it also includes a series of nonverbal communications. And I always challenge people to think about their nonverbal communication, how innate and hardwired it is. If you ever find yourself talking with somebody on the phone, you'll notice you're gesturing while you're talking. You'll notice there are other changes in how you smile, your facial expression, what have you. All those nonverbal communications are hardwired. Rationally, you know that the person on the other end of that phone has no idea of what you're doing. They're not watching you. They don't see you, but yet that's just part of our communication makeup. And it's so while individuals who might have depression or severe ADHD or severe anxiety might have other reasons they may not have reciprocal talking or things along those lines, the nonverbal communication impairment is really one of those things that we tend to hang our hat on symptomatically. It's a little bit more specific to individuals who have autism spectrum disorders. And then the final sort of sub area of this is really understanding relationships, differentiating what's a friend versus somebody I just go to school with, for example, understanding emotion and other people's perspectives, the capacity to put yourselves in somebody else's shoes, which we generally refer to as mentalization, are all aspects of social communication that we can see deficits for individuals on the spectrum. In addition, while social communication gets a lot of the press, another core area are restricted and repetitive patterns of behavior. And this is something that really is required for diagnosis and is also tends to be more specific to ASD. So that can include stereotyped movements, a need to keep the world sane, and some real pushback when there are perturbations against that need for sameness. There can be a series of sensory factors that come into play as well. And then sort of either unusual or overvalued interests. Somebody doesn't have to have all these, but they have to have some components of these to meet criteria for diagnosis of ASD. Okay. So now that we have a little bit of background around the biology and the symptom presentation, I wanted to talk about sort of where we're at with therapeutics development and where the needs and challenges lie. Okay. So this is sort of our punchline slide, which is really quite bleak. We have no approved medications for core symptoms in autism spectrum disorder. So you think of you'd be hard pressed, and I would even argue it would be impossible to think of another condition that affects 2.5% of the population where we don't have a medication that addresses the core disorder, whether it be diabetes, hypertension, depression, other psychiatric disorders. And in addition, ASD is unique among psychiatric disorders in not having therapeutics that have been specifically developed for it. Most other psychiatric conditions have some medications that target the core disorder. We have 2 medications that are approved for an indication of autism spectrum disorder, but they're specifically approved to treat irritability, which isn't a core symptom of ASD. I haven't talked about it up until this point because of that. It's an impairing symptom that some individuals with ASD may experience, and the medications that are approved can be quite helpful with respect to improving irritability and aggression. But again, that's a very small minority of individuals on the autism spectrum, and the approvals are also only in children, although we still continue to use those medications for individuals who benefit from them once they become adults. The approved medications that I just sort of passed through for irritability have associated with them a really broad series of potential side effects, and most people experience some of those side effects. And so we really have to be aware that the limited therapeutics that we have for the limited indication that we have carry with them some fairly significant downsides. Weight gain and constipation and sedation being the most common things that we see, which also can impact somebody's quality of life as well as have behavioral impacts too. There have been a number of challenges in developing medication therapeutics for individuals with autism spectrum disorders. One of the primary difficulties is that there's a lot of diversity for individuals who have ASD. And when you meet somebody with ASD, you've met somebody with ASD. You haven't met everybody with ASD, and that's because everybody has their own unique symptom presentation and experiences with the disorder. And so clinical trials often can be restrictive, or we try to target a population where we think there's a more homogeneous behavioral presentation, but then even if we show a response within that group, the generalization to the larger group of individuals with ASD may be more challenging. And so essentially, even if we develop a treatment that works for one specific subset of individuals on the autism spectrum, it may have no effect in another group of individuals. Concretely, we could think of something like, a clinical trial that looked at ASD, but individuals couldn't have seizure disorders, or they couldn't have a diagnosis of intellectual disability. Well, that would really impact how generalizable that could be to the wider community. We have a challenge with how we measure symptoms in autism spectrum disorders. We typically rely on reporting, usually of a support provider, a parent, or a family member, a spouse, friend, to monitor for behavioral change within a clinical trial. But many of those measures, again, may tap certain aspects of the condition. They may not more broadly as, say, ASD as a condition, and so those measures may miss some symptoms in some individuals with ASD, and since they're reliant on more subjective reporting as opposed to objective biomarkers. So a big push that we have in the community is to develop, in the scientific community, is to develop more objective ways of measuring change with respect to measuring brain waves or brain functional architecture through MRI and things like that. And then the final thing that's really challenging and often surprising to people is that clinical trials in ASD have a very large placebo response, meaning that people who are randomized to take a pill with no medicine in it often get a lot better when they enter into clinical trials. And there are a lot of complex reasons why that's the case, and a lot of reasons that we don't quite understand. But when we see high placebo response rates, when 20 or 30% of the placebo group in a clinical trial is improving significantly, that makes it incredibly challenging to differentiate that statistically from the treatment group. And so that's another major challenge that we have. And part of the reason why we may have that is because people have a lot of hope for improvement, and hope is a wonderful thing, but it can also cause what we call an expectation bias, that when people enter into clinical trials, they really feel like they're going to get better. And feeling that way, they actually make some changes in the positive direction. So we like to see people feel better and get better, and that's a great thing of entering a clinical trial. But as far as the scientific mission of differentiating people who are assigned to placebo versus the active medication, it becomes more challenging. Okay. Just to highlight some quick things, the reason that we lack therapeutics in ASD is not for a lack of trying. There have been many clinical trials addressing co-occurring symptoms, core symptoms, associated symptoms. I wanted to just show a few of the results. This is a clinical trial of kids where looked at a medication to see if it could help, a very commonly prescribed medication, to see if it could help with repetitive behaviors in autism. And you can see on the figure, essentially the progress for the group that was randomized to the medication citalopram, which is the black squares, is almost exactly overlaid for the placebo group. There was no difference at all. Secretin is a hormone that was looked at in many clinical trials in ASD, about 6 or 7 clinical trials, and there was a very nice review of that where those data were merged and looked at, and secretin didn't pan out despite significant hope and effort. This was a study of a medication called memetine, which is approved for certain types of dementia. And in this trial, again, the part of the trial that really matters is the part to the left of the dotted line on the figure. That's where it was a randomized trial and blinded, where people didn't know. Everything to the right is an open trial where people knew they were taking the medication. But you can see in the randomized portion, there's no real difference. Those lines are pretty much drawn over each other. This was a recent, really tragic failed initiative that we were involved in pretty heavily, which was a therapeutic called balovaptan, which looked at, which modulated a social hormone vasopressin. And there was an initial positive clinical trial in adults, but a replication of that study in a second group of adults, as well as a pediatric study, failed to show any treatment effect, and so that therapeutic was abandoned. These are some of the data that were from the pediatric side of that study, and you can see on the right, those are just basically how many, from a general impression, which we call the CGI, how many kids improved, and you can see it's basically those bar graphs overlap between the individuals who are in placebo, which is the blue, and the individuals who are on balovaptan, both at 12 weeks and 24 weeks, the balovaptan being the gray. And then on the left is the main outcome measure, which was an adaptive behavior scale, which showed no differences as well. So it's really quite demoralizing in many ways that we have many individuals who are on the autism spectrum who may really benefit from therapeutics, and we just don't have anything to offer. So the push has been to think about, instead of sort of serendipitous medication discovery, to think about biology. What are some of the shared common mechanisms that we consider are related to the disorder, and can we target those shared mechanisms to see if we can develop therapeutics for those specific areas? And so while we're still working on figuring out the biology of ASD, and at the end of the day, it's almost certain that there are going to be several types of ASDs that have a shared downstream behavioral profile or symptom cluster, but in fact are driven by different biological factors. So we're going to talk about 3 of those systems that come into play. The first is going to be the endocannabinoid system. So our body actually makes its own cannabinoids and has its own very complex hormonal system around this. That's why when people take cannabinoids from outside their body, we call that exogenous cannabinoids, we actually feel an effect in our behavior, physically, and the like, and that's because those external cannabinoids are actually flooding and overactivating the system. But the system is active throughout development and active throughout the lifespan, And as I said, it's a relatively complex system. Not going through all the different mechanics here, but this is just highlighting that there are a series of different regulatory factors in the cell and transcription of DNA and what have you that are responsive to the naturally produced endocannabinoids in the body. So these are some definitional aspects of it. The endocannabinoid system has a very important role in regulating of our immune system, how we perceive pain, how we regulate our mood, how we're motivated both socially and in other areas, our general socialization and anxiety. And so those are all important reasons why we look at the endocannabinoid system for individuals with ASD as a candidate site for therapeutics. There are several receptors that endocannabinoids work on. There are many different endocannabinoids. Some are more active than others. I have some of them listed here. And then, of course, things like THC, which is an exogenous cannabinoid, can also exert effects on these receptors. And that's actually how these receptors were initially identified. So there's clear data to show that changes in the endocannabinoid system can affect brain waves through EEG, and that they're associated with social play and anxiety in animal models, regardless of other factors. In individuals who have ASD, about a third have decreased expression of cannabinoid receptors when compared to community populations without ASD. And changes in different types of receptors, variations in those receptors, affect social reward and social processing. We've had a series of preclinical models, animal models that look at behaviors that can be somewhat mapped onto ASD, and endocannabinoid modulation can reverse and alter some of those behaviors in a positive way. And obviously, endocannabinoids can affect how neurons connect, and we call that synaptic function, which is something that's been consistently demonstrated to be disrupted in development for individuals with ASD. So there's been a pretty well publicized initiative to look at cannabidiol and THC in treatment of ASD, and a group out of Israel has really spearheaded this. And the main paper is cited here, where they looked at taking teens and young adults and kids who had ASD and randomizing them to either getting placebo or getting either a whole plant extract containing cannabis and THC, or pure cannabis and THC that had been refined. And they looked at some outcome measures for autism spectrum disorder, and the findings were really quite striking, which was to demonstrate that the people who were randomized to take the whole plant extract had very significant improvements in behavioral challenges that they were having, typically irritability and impulsivity, compared to placebo. You can see, as I said, the placebo response rate is still about 20%, so it's still quite high, but the actual clinical response rate to the therapeutic was about 50%, so it was so high that it still continued to separate statistically. But those studies haven't been really replicated. There are efforts right now to do so, so I don't recommend jumping to those interventions until we have the science. But there are efforts to continue to look at the endocannabinoid system to see if we can use that to help some individuals on the spectrum. And so the study that we're doing in collaboration with Janssen Pharmaceuticals is not, it works on the endocannabinoid system as well, but it does not include giving of external cannabinoids, as was done in that Israeli study where they gave cannabidiol and THC. In this investigation, we're actually slowing the body's breakdown of its own naturally produced cannabinoids. So essentially trying to turn the volume up a little bit on on the cannabinoid system in the body to see if that exerts an effect and a positive effect. The other nice thing that we're doing in the study as well is we're also looking at biomarkers. So people who come in for that study get to, watch certain types of social videos and biological motion videos, and we measure their brainwaves and their responses to that to try to see, even if there's an immediate clinical response during the short duration of a clinical trial, do we see other biomarkers of improvement as far as their brain development is concerned that they may not be aware of at that point. So the medication itself appears to be very well tolerated, at least when we've looked at people who've taken the placebo versus the pill with the active medication. There are a couple of nuisance side effects that have been reported, and these are the age ranges that we have in the study, which is 13 to 30. And the goal, all these studies are multi-site collaborative studies, so we're one of several academic sites working together to complete the study, and the goal is to have about 120 people. We're actually pretty close to finalizing. I think we have about 10 or 15 more people to close out the study. So we're anticipating the study is going to be done certainly by summer of 2022. Okay. Another area of investigation is in neuronal excitation and inhibition. So that's sort of a fancy way of saying brain activity, right? So there are different types of neurotransmitters in the brain. There are neurotransmitters that sort of excite the brain, and there are neurotransmitters that help calm the brain. And the two main ones are GABA and glutamate, GABA being the calming inhibition neurotransmitter and glutamate being the excitation neurotransmitter. And they do the heavy lifting with respect to that in the brain. There's a lot of data to support that some individuals on the spectrum have a relative imbalance of these two systems with a preference to excitation, and I guess that makes some sense clinically, right? Where sometimes people can get sort of absorbed in an area of restricted interest, or they might be more sensitive to sensory stimuli in the environment, and so there's a lot of interest in looking at how we can maybe help with medications to get a better balancing of the inhibition and excitation for individuals with ASD. So the real target is GABA at this point rather than glutamate, and there are reasons for that, but the main are that there have been a series of studies that have shown that for some individuals on the spectrum that GABA is actually, as far as the expression and genetics of it, can be disrupted in ASD. And several genes that have been implicated in ASD directly transcribe and produce GABA in the brain. So GABA is a complicated neurotransmitter, and the receptor system for it is quite complicated. There are, but essentially, activation of GABA receptors hyperpolarizes neurons. That makes them less likely to fire. So many therapeutics that work on GABA have a role in treating things like epilepsy, which is also quite co-occurring with ASD. Okay. So we sort of went through a good amount of this just to highlight the seizure aspect. Even though seizures occur in the minority of individuals with ASD, they still occur in about a quarter of individuals with ASD relative to 1% of the population otherwise. So we really have to think about this excitation side of the brain, where seizures are sort of a very concrete manifestation of a brain that's more prone and vulnerable to excitation. There are a number of genetic conditions that affect GABA and that are also associated with autism, including Angelman and Prader-Willi syndrome, and then we have a whole other slew of unnamed conditions where GABA synthesis, production, expression has been implicated in ASD. When we take pictures of the brain functionally and through something called MRS, magnetic resonance spectroscopy, we can see that there are differences in GABA expression between individuals with ASD and without. And animal models of ASD, again, it's always a tough concept thinking of how an animal model of ASD would really work. We could talk about that, but anyway, needless to say, animal models of ASD have also demonstrated changes in GABA. One of the subunits of GABA, which is the alpha 5, is really preferentially localized in areas that are really important for ASD in the brain. So there are different types of GABA receptors that are expressed in different areas of the brain. But the ones with the alpha-5 subunit are in the learning centers of the brain, in the motion regulation centers in the brain, and in the social areas of the brain. And so we have a mapping not only of other genetics, but anatomically seeing the expression of GABA in areas that are important for ASD. And so there's been a real effort to say, can we develop therapeutics that specifically work on the GABA-A alpha-5 subunit, GABA? And the reason is because you want that selective balancing in those regional areas rather than tickling other GABA receptors that could add sedation or other types of side effects, decreased alertness and what have you, that can happen with things like general benzodiazepines or even alcohol. So our study looking at this, which is in collaboration with Roche Pharmaceuticals, is specifically looking at what we call positive allosteric modulator. It's a fancy way of saying it activates GABA, specifically the ones that have the alpha-5 subunit in those areas of the brain that we think are relevant. This study has just started. The goal is to have about 105 people nationally. We have about 20 nationally at this point. And it's a randomized clinical trial where we have people randomized to either take placebo, low dose, or high dose. So it's a 1 in 3 chance that somebody would take placebo in that clinical trial, and it's a 12-week trial. Okay. And the last thing I want to talk about is, which I find the most interesting of the three, is the gut. And so we often don't think about the importance of the gut, but the data are continuing to grow and to support that there's a strong gut-brain connection, and the gut has its own intestinal flora called the microbiome. So the gut has its own bacteria, and those bacteria some of the bacteria do good things, some of the bacteria do bad things, and some bacteria do both. And those things include digestion of foods, but also release of different types of byproducts. And those byproducts can actually have impacts on neuronal signaling and firing. And the other aspect is that within the gut, there are a tremendous number of nerves. So we sort of think about the gut as essentially a second brain in many ways. Okay? So these are just some definitional aspects that we went through already, the metabolites being those byproducts of the bacteria and fungi and viruses and everything that lives naturally in the gut. Okay? So it's really important to recognize that there have already been numbers of studies that show that gut health impacts basically the entire body and has been associated with many different types of conditions, medical, psychiatric. Clinically, GI symptoms are incredibly common in ASD. They're much more common than in the general population. Usually about half of individuals with ASD have some gastrointestinal challenges, whether it be constipation, unsettled stomachs, diarrhea. And so right out of the gate, we have a recognition that there are differences in the gut for individuals with ASD versus without. And even more interestingly, the severity of gastrointestinal symptoms correlates directly with the severity of ASD in some studies, meaning that the more GI symptoms an individual has, the more likely their ASD symptoms are to be severe and vice versa. Okay. If there have been studies that have looked at the actual gut microbiomes directly for individuals with and without ASD, and it's quite clear that at group level, individuals with ASD have different gut flora that make up the gut and different proportions of certain gut flora relative to other individuals without. And so this was a study in children comparing the children with ASD in the red versus children without in the blue. Okay. There's also good data to support that a hormone called zonulin, which which regulates how permeable the gut membranes are, how readily they take in these byproducts, these metabolites from the bacteria. That gut permeability is higher for individuals with ASD. That expression of zonulin is higher. So meaning that individuals with ASD from a gut perspective are more likely to absorb the byproducts that are created by the gut microbiome than somebody without ASD. Why this is the case is not exactly known, and it may be a hormonal feedback loop with the microbiome itself. But nonetheless, when studies have been done to actually measure zonulin levels, that's been demonstrated quite definitively. So generally in the gut, we want to have a diversity of bacteria and fungi and things like that allow for a diverse community in the gut microbiome, but we can, and that we call homeostasis, but we can get dysbiosis. That's where things kind of get off kilter. That can be, we might have expansion of one particular type of bacteria, or that will crowd out others. We might have a reduced diversity, or we might have loss. And so there general population decreases across the board. And so those are different ways that the gut can be affected. There are many byproducts, as I said, many metabolites from the gut microbiome, but several have behavioral effects. Quite clearly, there are two that are here, which are P-cressyl and 4-EPS that are highlighted in pink. You don't really need to remember the names. Those are produced from food, right? So phenylalanine and tyrosine, those are dietary amino acids. Okay. Some of these metabolites have been looked at directly and been shown to both correlate with gastrointestinal and behavioral symptoms for individuals on the spectrum. And so a really cool thing that was done, which was published in really one of the 2 or 3 most prominent scientific journals about 2 or 3 years ago, was to take mice and to sterilize their gut from a microbiome perspective, and then transfer basically processed feces from kids with ASD and kids without into those mice, so to repopulate their gut microbiome with those children's guts, and then looking at the mice after that. And they actually looked at the mice directly, but then looked at transmitted, we call that vertical lines, rat pups and mice pups. There were mice pups, and then their children. And so the first thing that was demonstrated was that the actual types of bacteria in the gut of those mice varied. That would make sense since they were since we know that kids with ASD have different gut microbiomes than kids without. So that wasn't a surprise. But the behavior of the mice varied too. So the mice that received the gut transplant from kids with ASD were more likely to be repetitive in their behaviors. They were less integrated, less social, and less willing to explore their area than the mice that got the transplant from neurotypical children. And so, and remember, these are all the same type of mice and had gone through it. So the only difference is from which kids they got the transplant. So it's really not to say that the mice have ASD, but the behavioral profile of the mice really did vary at group level. Okay. And then this is another way that they showed it with correlations as well between different types of bacteria. Okay. So they were able to show that different concentrations of the bacteria correlated with different behaviors in the mice. Okay. So the final therapeutic we have is in collaboration with Axial Pharmaceuticals, and it looks at this specific issue. So there's a nanoparticle bead. It doesn't get absorbed into the body, it stays in the gut, but it acts like a sponge and it sops up some of these metabolite byproducts of the gut microbiome, the ones that we think may be associated with certain symptoms in ASD. This type of medication is already used for individuals who have chronic kidney disease because it allows absorption of those byproducts and in the context of kidney disease, they can't clear it through their kidneys. So it sort of sequesters out. But we've been looking to apply it for individuals with ASD. So this is what it looks like. This is on an electron microscope. These are incredibly small beads, and they're porous. So small metabolite chemicals can get combined to these beads and then just be eliminated in the feces without getting absorbed back into the body. Obviously, it doesn't go into the bloodstream at all, so it's actually really excellent from a safety perspective. Some people might have some abdominal bloating, things like that, but generally that's not even reported. And so really has minimal risks and side effects. The one thing we worry about is if people are taking medicines, could the medications themselves get absorbed onto those beads? But we have ways of working around that too. Okay, so that study that we're doing is in teens, and we're really excited. We just started that study within the last few weeks. Okay, but in summary, so I think the take-home points here are that ASD is a really complex condition. There are a lot of genetic and biological factors. There's not much on the parenting side, so it's important to remember that. This is really a biological disorder, and we have to be really mindful of that. And all the more reason why developing biological therapeutics makes a lot of sense. But we've had a lot of challenges in doing that. The diversity of the condition is one of those factors among many. So we really have to have a neuroscience-informed approach. We really have to continue to pound on understanding mechanisms and developing hypotheses for shared mechanisms of the condition so that we can do targeted therapeutics for those. So this really requires a lot of collaborative work, not only between the ASD community and their support providers, but with clinical providers, research teams, and broad research teams, bench research teams looking at the very most simplistic level of compounds and chemicals, all the way up to bedside where people are engaging in clinical therapeutic trials and trying to advance for the entire ASD community. And then just a little plug, if anybody's actually interested in working with us as a community partner, as a participant, either in our clinical trials or in our studies of neurobiology of autism spectrum disorders, please feel free to reach out to us. Don't hesitate to reach out to us. Alexis Laval is our team coordinator and program lead, so she would be the person to reach out to. So we'll end there, and I just want to thank everybody for watching.